Hey guys, today we're going to talk about mitosis. I want to quickly review over some terms that you'll see when talking about DNA. All of this that you see on the board right now is DNA, it's just in different forms. Typically, in a typical non-dividing cell, you are going to see the DNA look like this, in single-stranded, uncondensed form, and that is called chromatin. Now, if a cell is ready to divide, it's going to copy its DNA. When it copies its DNA, it's going to make an identical copy to itself. These two are called sister chromatid. Okay. And then once these sister chromatid condense and pair up with each other, it's going to form this nice looking X shape, which we call a chromosome. Okay, so chromatin, sister chromatid, and chromosome is all DNA. It's just different terms we use when we are looking at them in different forms. So chromatin is when it is uncondensed in a non-dividing cell. When the cell's ready to divide, it's going to make a copy of its DNA. That copy is now called the sister chromatid. And when those copies pair up with each other, they form what we call the chromosome. Okay, so we're looking at a cell that has 4 chromosomes. So its 2n number would be 4, and its n number would be 2. So this is going to represent our cell membrane, and we're going to be looking at an animal cell. So I drew it in a circular shape. And then here is our nuclear envelope. We aren't going to worry about looking at any of the other organelles. We just kind of want to focus on chromosome movement. So in interphase, there are 3 major steps. You have the G1 phase, which is just for growth. Inside of the nucleus at the time, you are going to have a dad copy of chromosome 1, a mom copy of chromosome 1, a dad copy of chromosome 2, and a mom copy of chromosome 2. Okay. These are not going to be visible under a microscope at the time. They're not condensed, so you won't see them, but this is what you would have in this typical nucleus during G1. In S phase, you are going to have DNA replication. So we are going to take these chromatid and make a copy of them. So we are going to copy chromosome 1 from dad. We are going to copy chromosome 1 from mom. We are going to copy chromosome 2 from dad. And copy chromosome 2 from mom. So in the S phase, we have our sister chromatid, and they have been replicated. In G2, you're going to have another series of growth, and then the cell's going to check and make sure that it's ready to divide. If it is, then this cell's going to start the phases of mitosis. And mitosis is nuclear division. So we are going to be dividing the nucleus in mitosis. So the first phase to that is what we call prophase. In prophase, these replicated DNA strands are going to condense down into chromosome form. So they're going to take their X shape, and they are going to start to become visible under the microscope. Your nuclear envelope is going to start breaking down, so that DNA can come out of the nucleus, and then we would be able to separate it. You are also going to have the start of the formation of organelles that are only found in animal cells. They're called centrioles. These centrioles are going to form what we call spindle fibers. And spindle fibers are going to help move these chromosomes during the rest of the phases. Okay, so that's prophase. In the next step, which is called metaphase, I'm going to erase this. In metaphase, our nuclear envelope is completely gone. We still have our centrioles. Now our chromosomes that we have made are going to start to line up in the center of the cell. This center of the cell is called the metaphase plate. Spindle fibers from the centrioles are going to attach themselves in the center of each of these chromosomes. And like I said before, these spindle fibers are just going to help pull those chromosomes apart. Okay, that's it for metaphase. Okay, they line up in the middle along the metaphase plate. Spindle fibers attached to the center here. This is the longest phase of mitosis. There's going to be a checkpoint that the cell's going to go through to make sure that these have lined up correctly. Once the cell has shown that it is correct, it's going to move into the next phase, which we call anaphase. In anaphase, these spindle fibers that are attached at the center of the chromosomes are going to shorten up. And as they shorten up, these chromosomes are going to start to split apart and move to what we call the pole of the cell. So they're going to separate from one side to the other. That's anaphase, pretty simple. The next and final step of nuclear division is what we call telophase. In telophase, we are going to have the nuclear envelope reform around both sides of our DNA that we just split. Centrioles are going to disappear. So here's my nuclear envelope reforming around there. Here's my nuclear envelope reforming around these ones. Chromosomes are going to start to decondense and become less visible. And the last and final step, not part of mitosis, it's a separate step, because we've already divided the nucleus. We have to divide the rest of the cell, and that step is called cytokinesis. All that's going to happen is the cell membrane is going to start to pinch along the middle. In an animal cell, this is called a cleavage furrow. That cell membrane is going to pinch in the middle, and in the process of doing that, it's going to divide the cytoplasm, and it's also going to separate all of the organelles that you would find floating in the cytoplasm. And we are left with two identical daughter cells that have the 4 sets of chromosomes that the mother cell started off with.